Hello everyone. So the next topic which we will be going to discuss is about the C4 pathway. So this pathway is also known as the Hatch/slash pathway. Why this is known as Hatch/slash pathway? Because this C4 cycle was being discovered by Hatch and Slack. They were the scientists in Australia in 1966. So that is why this pathway is also known as Hatch/slash pathway. So let's start with the introduction first. We have already discussed this part. So this pathway was named after the discoverers, that is the Hatch and the Slack pathway. Now, if we talk about C4 cycle, in some of the plant species, there is an alternate mode of carbon fixation that have evolved. Why they have evolved? Because they have evolved to minimize the photorespiration and optimize the Calvin cycle. So the most important of these photosynthetic adaptations are C4 photosynthetic carbon cycle. So this cycle, the C4 cycle, they concentrate CO2 at the site of carboxylation and they keep the CO2 concentration high enough for the rubisco to bind the carbon dioxide rather than the oxygen. So what is happening in this pathway is, in this pathway, C4 cycle have evolved to minimize the photorespiration. So there are some plants that have C4 cycle. So that is why these plants are called as the C4 plants. So in these slides, we will be discussing some of the examples of C4 plants as well. So every photosynthetic plant, they follows Calvin cycle. But in some plants, there is a stage to the Calvin cycle which is known as the C4 pathway. So this pathway, here is a 4 compound, C4, which means there is a 4 carbon compound. So this pathway is also known as the oxaloacetic acid. And it is the first product by the carbon fixation. So that is why it is also known as the oxaloacetic acid. Now, this is a pathway, C4 pathway, in which we are representing two cells here, that is the mesophyll cell and the bundle sheath. So, what is happening in this cycle is, in the first step of this cycle, there is phosphoenol pyruvate. There is a phosphoenol pyruvate. So, this is mentioned here. It is carboxylated to oxaloacetate. That is, oxaloacetate is considered as the C4 acid in the cytosol of the mesophyll cell. So this is representing the mesophyll cell. So that is why the process of C4 pathway, one of the process of C4 pathway takes place in the mesophyll cell. And this is the first product of photosynthesis. So C4 or oxaloacetate, this is the first product of, it is the first product which has been formed by the photosynthesis. In contrast, 3-phosphoglycerate in the C3 cycle. So there is a 3-phosphoglycerate which is formed in the C3 cycle. So this reaction is catalyzed by PEP carboxylase. Why? Because there is an initial CO2 fixation product is a 4 carbon containing organic acid which is named as the C4 cycle, and oxaloacetate, it is then reduced to maltate. So oxaloacetate is then reduced to maltate in the chloroplast of the mesophyll cell. So it is being the process is being taken place in the chloroplast of the mesophyll cells, and the maltate ions, they are then transferred through the plasmodermata to the bundle sheath. So this is being transferred to the next part of the C4 pathway, that is the bundle sheath, and they are decarboxylated here. So this generates a very high concentration of CO2. So there is a very high concentration of CO2 within the bundle sheath. So high concentration of CO2 in the bundle sheath. And rubisco, it is confined to the bundle sheath cells. Now the CO2 concentration which is in the bundle sheath is sufficient to suppress the oxygenation activity of rubisco. So it will basically suppress the activity of rubisco, and the CO2 is fixed by rubisco and it is converted to triose phosphate by the Calvin cycle. So C3 is known as the Calvin cycle. A C3 acid pyruvate returns to the mesophyll from the bundle sheath, and in the mesophyll cell the pyruvate is converted to phosphoenol pyruvate, and this step is ATP dependent. So C4 cycle is therefore an ATP driven CO2 enrichment cycle. So if we consider the C4 pathway, what is the C4 pathway, C4 cycle? It is an ATP driven enrichment cycle. The cost of concentrating CO2 within the bundle sheath is 2 ATP per CO2. So that is 2 ATP per CO2. And if we consider total energy which is required for fixing CO2 by the combined CO4 and the Calvin cycle, it is the 5 ATP plus 2 NADPH per CO2 which is fixed. So in fact, what is happening is C4 cycle, it concentrates CO2 in the bundle sheath, keeping the CO2 concentration in the sheath high enough for the rubisco to bind to carbon dioxide rather than the oxygen. So in this way, C4 photosynthesis minimizes the photorespiration and enhances the carbohydrate production. So this is what is happening in the C4 pathway. So what it does, it minimizes the photorespiration. So in the first slide we already discussed that the C4 cycle minimizes the photorespiration and it optimizes the Calvin cycle. So this is what is happening here also. C4 photosynthesis minimizes the photorespiration and it will enhance the carbohydrate production. Now this is all of the summary which I have already explained to you. Next is what are the examples of C4 plants. So the examples are corn, sugarcane and some of the shrubs. So these are the examples of C4 plants. Next, C4 plants, they there is a two distinct types of photosynthetic cells, that is the bundle sheath and the mesophyll cells. In the diagram that was being discussed, we have discussed this, that this was the mesophyll cell and this is the bundle sheath cell. So we have already discussed this. Now in bundle sheath and mesophyll cells, there are 5 stages which are presented. So these are the 5 stages in both the mesophyll and bundle sheath cells. So first is there is a fixation of carboxylation of phosphoenol pyruvate in the mesophyll cell, that is to form the C4 acid. It was being depicted in the diagram as well. Then oxaloacetate then converts into other C4 acids such as maltate and aspartate. There is a transport of C4 acids to the bundle sheath. There was in the diagram below, this was the mesophyll cell and this was the bundle sheath cell. So this process is showing exactly what is happening in all the 5 stages. So this is the basic 5 stages of the C4 cycle. Next step, the fourth step, which is the decarboxylation of C4 acids within the bundle sheath and regeneration of the CO2 molecules. Then the transport of C3 acid, that is formed by decarboxylation step back to the mesophyll cell. So this is what is happening in the C4 cycle. Now this is a leaf anatomy in C4 plants. So as we have already discussed, the photosynthesis in C4 plants takes place in two types of cells, two distinct types of photosynthetic cells, that is the mesophyll cells and the bundle sheath cells. So C4 plants, they are very unique in processing these two types of photosynthetic cells. Inner ring of bundle sheath cells around the vascular bundle and the outer layer is of the mesophyll cell. So if we view a transaction section under the microscope, the ring of bundle sheath gives the appearance of word surrounding the vascular bundle, and this wreath-like arrangement of the chloroplast, which is bearing cells, which surrounds the vascular bundle is termed as the Crank's anatomy. That is the Crank's anatomy. So this is what is been observed in the microscope. Then mesophyll cells, they are connected to the bundle sheath cells in large numbers of plasmodesmata. Enzyme PEP carboxylase is present in the mesophyll cells, whereas Rubisco is located in the bundle sheath cells. So where Rubisco is located? Rubisco is located in the bundle sheath cells. Rubisco. In addition, mesophyll cells contain chloroplast with stacked thylakoids. So mesophyll cells contains the chloroplast. It contains the chloroplast. Chloroplast dimorphism occurs in the C4 plants. And Crank's anatomy is not present in all the photosynthetic organisms, while C4 cycle is present. So in few land plants and diatoms, there is a complete C4 cycle which occurs in a single photosynthetic cell. So that is why this is the mesophyll cell and bundle sheath cells, they are being represented. So these are all the references